Hi everybody, I'm Ben, and in this video I'll be explaining a little bit about my research project regarding GAA microsatellites and their involvement in cancer. So to start off with a little bit of background information about the research project in general and the hypothesized transcriptional mechanism that we'll be investigating. You can take this as just a representative of a stretch of DNA. And to give you a little bit of background on GAA microsatellites, a GAA microsatellite is 1 to 10 base pair of nucleotide repeats with specifically the GAA sequence. So you can see here that this would be a 1 to 10 base pair stretch of GAA. And then an oncogene is any gene that can conditionally transform cells to malignant tumors. So based on the cellular conditions, an oncogene can transform a cell into cancerous. So the Weigel lab has been investigating transcription factor activating protein 2A, or TFAP2A, and how it functions through these microsatellites. So TFAP2A does not bind to a GAA consensus sequence, so we believe some complex is recruiting TFAP2A to the microsatellite. And once it's recruited here, we're seeing repression of these oncogenes within 50 kilobases of the microsatellite. So further, sumylation is a small ubiquitin-like modifier that gets added onto proteins. It's a post-translational modification. So what sumylation does is it adds this sumo group to TFAP2A and it blocks the repression through this microsatellite. So why would this be important? Well, the importance of this hypothesized mechanism is that if sumylation is dysregulated, then you can see blocked repression of these oncogenes, and if you don't have repression of the oncogenes, you'll see roughly a 2 to 4-fold increase in the oncogene expression. And that can lead to increased dedifferentiation and a more highly metastatic melanoma. So, essentially, if you can control this pathway, this dysregulated sumylation, you could decrease melanoma incidence and also metastatic aggressiveness. So in goal 1 of this project, I want to delete the microsatellite from an experimental model of A375 melanoma cells. That is where the Weigel lab has observed this unconventional TFAP2A recruitment to this GAA microsatellite. So I'll use A375 melanoma cells as a model, and I'll do a lipofectamine transfection where I will introduce GFP, CRISPR-Cas9, and guide RNAs into the nucleus, where they can create a double-stranded break guided by these RNAs, which will lead them to the sequence of the GAA microsatellite. And after the double-stranded break happens, GFP will be present, and post-transfection we should see incorporation of the GFP into the DNA sequence and deletion of the GAA microsatellite. So that's how I plan to delete the microsatellite, and to test for that, I have to select for positively transfected cells, so cells in which the transfection was successful. And I'll do that by using flow cytometry and sorting for positive GFP cells. So the flow cytometer uses electricity to sort cells into single cell colonies, and it can sort them based on positive or negative GFP expression. So then I can take those single cell colonies, expand them, and duplicate them, so then I can grow out cells in duplicates, and I can harvest DNA from one of those duplicates and run gel electrophoresis on it. So these would be DNA bands from the cells that I harvested. And if we evaluate that a little further, the red highlighted band here would be wild-type DNA, still containing the GAA microsatellite. The yellow highlighted band would be a band of DNA in which the GAA microsatellite was actually deleted. And you can see it's a smaller fragment because it traveled further, it migrated further during the electrophoresis. So since it's a smaller band, it's a suspected deletion. So what we would do is we would excise this band from the gel, harvest the DNA, and submit it to the core to do Sanger sequencing, and they can give us a result and either confirm that the GAA sequence is missing and deleted, or not, and we'll go back to the drawing board. But if it is deleted, we'll proceed to goal number 3. So goal number 3 is to investigate the effects of the deletion, and we'll do this by using real-time quantitative polymerase chain reaction, so RT-qPCR. RT-qPCR is going to measure gene expression, specifically transcription, and transcription is the process where DNA is converted to RNA, and then later translation will convert RNA to protein. So transcription is the process we're interested in here. So how does this work? You start off with your wild type A375 melanoma cell. This is my model. We do goals 1 and 2, get the deletion and confirm the deletion. Now we have a mutated A375 cell missing the microsatellite. So then what I would do is I would run a QIAGEN RNA-seq kit to harvest the RNA from that cell. And this RNA is going to be the transcription of the DNA of this cell. So then what I can do is I can run reverse transcription, which would make complementary DNA sequence. And with that complementary DNA sequence, I can run RT-qPCR. So RT-qPCR is the process, the biological assay that I'll use to investigate this expression. And it's very important, so I'll explain a little bit further how RT-qPCR works. So in RT-qPCR, you start with your DNA sequence. This is your 5' to 3' and 5' to 3' this way. So this is your cDNA sequence, and you're going to have your forward PCR primer and your reverse PCR primer. These are just going to be DNA templates that match these sequences and recruit DNA polymerase so that it can start copying the sequence. Now for qPCR, for the quantitative aspect, you need what's called a probe, and in the probe you'll have a fluorophore and a quencher. The fluorophore actually is fluorescent and it emits fluorescent light, but when it's attached to the quencher actually absorbs the fluorescent light and none is emitted from the fluorophore. So once you begin the amplification, you'll get DNA polymerase II, which will come and start amplifying the sequence. And as soon as DNA polymerase actually hits the probe, it will cleave the fluorophore from the probe. And at that point, when it's released from the probe, the quencher can no longer quench the fluorescence, and it will actually be emitted at that point. So you'll get 2 products. You'll get your replicated cDNA, due to the copying of the sequence by DNA polymerase, and you'll also get fluorescence as these products are created. So what you can do is you can read that fluorescence and generate what's called an amplification plot. The amplification plot depends on the concentration of the cDNA. So the higher the concentration of cDNA, the faster you'll see that it amplifies, and the lowest concentration will amplify the slowest. So you can use this information to determine how much cDNA is present. And why is this important? Well, if you remember, the cDNA was generated from the RNA, and the cDNA amplifies based on how much there is. So basically this is just a measurement of how much RNA was made from your mutated A375 cell. So it measures the gene expression or transcription of that cell's DNA. So what are the expected results if our experimental hypothesis is true? Well, in wild-type A375 cells, the positive control condition, you have relative oncogene expression as the y-axis here, and a SUMO inhibitor in one condition. Now, if you recall, SUMO inhibitor would actually restore repression of the oncogene. So you would see a 2- to 4-fold decrease in oncogene expression compared to, a no treatment condition. In a negative control, the difference would be is that you would use not wild type cells, but TFAP2A knockout cells. And in those cells, you would expect no TFAP2A to be present here, so you would expect that repression will not occur through the microsatellite regardless of sumo inhibition or non-treatment. And in the experimental condition, you would run sumo inhibitors for both control trials, but you would have in one condition a microsatellite deletion. So essentially you would have TFAP2A present, you would have sumo inhibited, but you would remove the microsatellite, and we expect oncogene expression to increase 2 to 4-fold, and that would support the hypothesis that this microsatellite is necessary for repression of these neighboring oncogenes within 50 kilobases. So what does this mean? Why is this important? What is this project going to mean? So just a quick recap, TFAP2A would be driving oncogene repression through these microsatellites. So the microsatellites, once they bind TFAP2A, would repress oncogenes, and uncontrolled sumolation would inhibit TFAP2A. So it would actually reduce this repression, and those oncogenes would increase in expression, and you would see those cells begin to form tumors that are cancerous. So bottom line, if you can control for this sumolation at GAA microsatellites, you could reduce melanoma occurrence and metastatic capability.